cell, then it becomes semi-autonomous. It involves the proteins, and the proteins they were called as FTS, and they were similar to those that mediate the cell division in bacteria. So these are the certain you can say that evolutionary footprints or the things which can confirm that they are that the particular cell organelle was bacteria actually, and it is embedded into the cell. So certain kind of characteristics is still retained by that particular organelle. One of them is the FTS protein that helps in the division of the bacteria. Cyanidiocyzone, that is the chloroplast dividing. Here you can say that you can see that how the chloroplast is dividing. It's very easy to understand the thing. One of the example is the parasitic plant genus Rafflesia, which is obtained its nutrients from other plants, specifically uh, stigma vines. Since the Rafflesia, this particular plant, Rafflesia, you have learned a lot during the course of your study from 11 to 12. And Rafflesia is considered to be the to have the biggest, or you can say the largest flower that about 100, that about 1 meter in diameter. So it's in the Rafflesia, the flower is almost about 100, you can say that 100 centimeter in diameter or 1 meter in diameter. The pollination which occurs in case of the Rafflesia that is carried out by the foot of the elephant, and the smell is very bad. In case of Rafflesia, it's similar to the rotten meat. So this is this plant is comparatively, you can say that very known when we were discussing about the botany. Which one is the smallest plant? Which one is the smallest flower? And which one is the largest flower? So in that way, this Rafflesia is very well known. So you can say that if we are taking the example of the Rafflesia, since Rafflesia gets all of its energy from parasitizing any another plants, it no longer needs its chloroplast and has lost the genes coding for the development of the chloroplast over a long period of the evolutionary time. Rafflesia is the only genus of the land plant to be lacking the chloroplast. The chloroplast genome. Chloroplast genome, this is called as the cpDNA. As I said that the DNA, the independent DNA is present in case of the chloroplast, the proteins or the ribosomes, they were present. So the whole machinery, almost whole machinery is present, which can helpful in the multiplication and also in the survival of the particular organelle. So chloroplast has its own genome, and that means the DNA, and this is called as the cpDNA. That is chloroplast DNA, and this is also known as the plastid DNA or the ptDNA. It is a circular double-stranded DNA molecule. When we are talking about the chloroplast genome, the chloroplast genome size ranges from 120 to 217 kb, with the majority of the plants fall into 120 to 160 kb. So you can say that it's not very long DNA. You can say that 120 to 117 kb means kilo base pair. So it's not very long, but in comparison to the higher plant, it is definitely it's a long one. But whenever we are comparing when we are making the comparison between the um, eukaryotic DNA, and in case of the chloroplast gene, it's comparatively very small. So, as in case of the Pelargonium, has the chloroplast genome size is 217. Yeah, it contains about 100 genes to synthesize protein, so it's not a very small one. You can say that, but because 100 genes, it bears the 100 genes. So 100, you can say that 100 points were there where the messenger RNA can be formed. Because all of you know that messenger first, the messenger RNA were formed, and that messenger RNA were sent for the production of the particular protein through the ribosomes, and that ribosome can be helpful in the production of the proteins. So 100 genes were present in a small DNA molecule that is about 120 to 217 kilo base pair. The chloroplast DNA region includes a large single copy LSC, a small single copy SSC regions, and inverted repeats IRA and IRB. These are the different terminology which I'm emphasizing to you people, because they are in very much details. They were not included in the course, but at least you must know all these things. This is important thing, like, because some of you may go into the biotechnology or microbiology or some other higher studies. If you people go in some higher studies of biology or biochemistry, then all these things will be helpful in understanding the things. So at least the terminology you must know. The large single copy, the LSC, small single copy, SSC regions are there. Then inverted repeats were there. That is called as IRA and IRB. Then conifers and group of the legumes lack these inverted repeats. So in the intermediate plants, they were called as gymnosperms, these kind of things were not found. Then characteristics of the chloroplast genome. cpDNA is relatively abundant component of the plant total DNA, thus facilitating the extraction and analysis. The conservative rate of the nucleotide substitution enables to resolve plant pathogenetic relationship at deep level of the evolution. Example, mono and dicotyledonous. Then chloroplast protein coding genes involved at the rate that is on average five-fold slower than the plant genome, the nuclear genome. Strictly maternally inherited in most of the angiosperms, while in conifers it is inherited in paternal. The chloroplast DNA is passed on from one generation to the next generation with only an occasional mutation alteration, altering the molecule. Sexual recombination does not occur. As you have seen in case of the higher plants and animals, what happens during the meiosis, especially at the stage that is called as the protein and dikinesis. You can say that the pacatine, diplotin, and dikinesis. In these three stages, the exchange of the genetic material occurs. And during the exchange of the genetic material, what happens, the resultant will be, you can say that the DNA, if the DNA is changed, then definitely the phenotypic characteristic is also going to be changed, like skin color or a number of things. Even in case of the humans, you can say the face is also changed. So that kind of things are not at the lower level, especially in case of the bacteria, and we have considered that this chloroplast was initially was bacteria, you can say, and that bacteria were invaded into the cell. So therefore, the DNA in case of the chloroplast is also very primitive, you can say that, and even the replication is also the simple replication happens. There is no any kind of meiosis, or you can say that there is no any kind of um, genetic material exchange occurs in the two different cells. But sometimes due to one or the other reason, the mutation can be occur. So this is the information about the characteristics of the genome chloroplast. Then molecular systematics of on the cpDNA. Then cpDNA regions can be amplified by the means of the PCR. So most of the people said the PCR machine. Actually, PCR machine is not. It's a simple reaction that is called as polymerase chain reaction, and that polymerase chain reaction is meant for the production of millions of the copies of the DNA. अगर आपके पास कोई डीएनए है उसकी अब आपको मिलियंस ऑफ कॉपीज बनानी है तो वो आप इस प्रोसेस से बना सकते हो जिससे is chain reaction. And this polymerase chain reaction can be carried out by the particular machine. Some people call it as PCR machine. However, it is a thermal cycler. वो थर्मल साइक्लर होती है एक छोटी सी मशीन उसमें क्या होता है कि टेम्परेचर जो वेरी करता है वो बहुत तेजी के साथ वेरी चेंज हो जाता है कि आपको 90 डिग्री टेम्परेचर 95 डिग्री टेम्परेचर से अगर आपको 2 डिग्री पर भी लाना है तो विद इन सेकेंड्स वो टेम्परेचर 2 डिग्री पर आ जाएगा तो डीएनए को मल्टीप्लाई करने के लिए इस मशीन को यूज करते हैं सिंपल इट्स अ वेरी सिंपल थिंग एंड यस और इसमें जैसे डीएनए को 90 डिग्री 95 डिग्री टेम्परेचर तक ले जाते हैं और 95 डिग्री टेम्परेचर ले जाने के बाद में डीएनए के जो स्ट्रैंड हैं हाई टेम्परेचर की वजह से वो खुल जाते हैं जैसे ही हाई टेम्परेचर होगा उसमें तो वो खुल जाएंगे जब वो खुल जाएंगे तो जो उसमें आपको अलग से प्राइमर्स देने पड़ते हैं प्राइमर्स मीन्स कि उसमें आपका जो सिंगल स्ट्रैंडेड डीएनए होगा जिसमें आपके लाइंस वाला सब सपोज देन 10 बेस पेयर्स का होगा तो 10 KB का होगा 1 किलो बेस पेयर का तो 10 डिफरेंट बेसिस जुड़े हुए होंगे या 20 बेसिस जुड़े हुए होंगे वो इस तरह की हजारों कॉपी आप उसमें के मिक्सचर में डाल देते हैं तो जैसे ही वो दोनों स्ट्रैंड डीएनए के खुलेंगे जो कॉम्प्लीमेंट्री स्ट्रैंड होगा वहां जाके आपका जो प्राइमर है प्राइमर कहते हैं उसको प्राइमर जाके लग जाएगा और उसके बाद में जो एक एंजाइम होता है टेक डीएनए पॉलीमरेज जिसको थर्मस एक्वेटिकस से निकाला जाता है बैक्टीरियल जीनोम से और उसका काम ये है कि वो इतने हाई टेम्परेचर तक सरवाइव करता है वो फिर वो पूरी चेन आपको जो डीएनए की चेन रह गई है वो पूरी कंप्लीट कर देता है सो दिस इज द प्रोसेस ऑफ द पीसीआर एंड और लास्ट में हम जब ये जो साइकिल है जो टेम्परेचर बढ़ना फिर जो है आपका टेम्परेचर कम होना फिर एक्सटेंशन सपोज फर्स्ट स्टेप इट इज कॉल्ड एज द डीनेचुरेशन ऑफ द प्रोटीन्स दूसरा होता है अनीलिंग अनीलिंग मीन्स जो आपका प्राइमर है वो जाके लगेगा तो एट दैट टाइम टेम्परेचर कम हो जाता है 35 से 45 डिग्री के टेम्परेचर के बीच में आ जाता है फिर आता है एक्सटेंशन एक्सटेंशन में क्या होता है जो डिस्पेंड है कंप्लीट डीएनए का वो बनना शुरू होता है बहुत सारी आप बेसिस दे देते हैं जो आपके पास बेसिस हैं एडेनिन गुआनिन साइटोसिन एंड थाइमिन ये अलग अलग से दे देते हैं आप उसी मिक्सचर में तो जहां जहां जरूरत होती है उसकी कम्प्लीमेंट्री वो लगता चला जाता है और टैग डीएनए पॉलीमरेज उसकी पूरी चेन बनाता चला जाता है so it is very much important technique nowadays in case of the molecular biology whenever we are talking about the you can say that the molecular biology techniques. So because nowadays we are not able to carry out these experiment due to the pandemic. If it was there, then I can show you that particular machine, PCR machine that can help in the amplification of the DNA. That can be helpful in the multiplication or the amplification of the DNA. That particular PCR machine. The resulted PCR product may be subjected to the RFLP and or the DNA sequence. So different kind of DNA sequencing can be क्या कहते हैं DNA sequencing can be done. Then common chloroplast DNA region used in the systematic studies, RBCL 1400 base pair where they are TRNL, TRNF 250 to 800 kb base pair we are present, and ATPB. These are the different regions uh, of the chloroplast DNA, and they are very much helpful in the different kind of studies of the DNA. This is the whole genome of the chloroplast. You can see that number of things we are present, number of things, and number of identified regions we are present. So, if anyone want to cut this particular region and can want to add some other DNA sequences that can helpful and that can be helpful in this particular genome of the chloroplast. Number of things they are present. They are not very much useful for you people right now. So that's why I'm not going into the detail. Otherwise, you people will become confused on it. Then plastids. Plastids is another important energy using cell organelle found only in plants. Then Schimper coined the name plastid for those structures responsible for the photosynthesis. Actually, the photosynthesis provides the chemical energy directly or indirectly for all the other living organisms, and chloroplasts are the unique organelles. For that, they are capable of capturing, converting, and conserving the solar energy in the form of the chemical energy. Plastids are found in almost all the cells of the plant body, either in the form of the colorless plastids or the colored plastids or the proplastids. There are several forms of the plastids. The proplastids, they are the precursors of the plastids found in the meristem. Then etioplast, formed in the shoots of the dark grown plants, distinctive internal structures. Then chloroplast in all the green tissues. Then amyloplast, that is prominent in the roots, store starch and colorless. Sorry. Then chromoplast in mature fruits, lots of the carotenoids and little chlorophyll is present. So there are number of forms of the plastids which were found in case of the plants. This is the photo. This is the diagrammatic representation. How you can identify, like from the proplastids, then etioplasts were found, and the etioplasts and chloroplasts were also found from the chloroplast. Then from chloroplast, the chromoplast can also be found. And the leaf from the leucoplast, the amyloplast, elioplast, and the proteinoplast were formed. Then plastids development in dark ground barley. So how this is the oldest one, this is the youngest one, this is the mature ethioplast and the proplastid. So this proplastid can be grown into the mature ethioplast. You can see that the dark grown plastic is there, and the size is also increases from 1 micron to about 4.5 micron. And number of plastic cells, they may be from 0 to the 65. in case of the particular cells. Thank you very much. This is a few information regarding the co-plast regarding the plastics, and now we were able to complete this particular topic. Do you have any idea that today is the last day, I think, of the session? Anyways, on Thursday, I'm going to take the class, inshallah, in spite of the last working day. You haven't got any kind of the examination schedule right now. So I request you to please go through the different topics which is included in your course and complete. I think you have also completed the assignment and both the assignment, right, from the theory and one from the practical, because they carry a very big marks, right, from means 70% marks they will carry it out. So please be careful about that. That will be very helpful. Otherwise, most of the students who are not taking seriously, they may suffer because from 70 you will get some good marks, but right from the 30, if you are not able to answer the uh, questions properly, then definitely it will be a problem for you people. So please be careful, be confident, and start your studies right from the day one. अभी आपके पास टाइम है, अभी आप तैयारी कर लो बहुत अच्छे से, बाकी बहुत प्रॉब्लम होने वाली है. If anyone is facing any kind of problem regarding the topics, anyone can WhatsApp me on personally WhatsApp me, not on the group, because on the group there are a number of teachers we are included, and they may be facing some problem because every time the message is coming. So you can message me, you can WhatsApp me personally. That will be manageable, easily manageable to me, and you also, and that can also not disturb other teachers. Thank you very much, and all the best for your exam.
Excuse me, sir.
Yes, please.
Assalamu alaikum, sir.
Anything?
Sir, I have sent the practical assignment on your email, sir. But how we can know that it is reached, sir? Because there
If
is
it
no
is not,
message.
if it is not bounced, then it is expected that it is reached. Okay. If you have typed the email ID properly, and if it is not bounced, then definitely it will reach to me. Anyways, because as I said in my previous classes, that due to the number of classes I am dealing with, so almost all were completed, and the assignment marks were also finalized, except the MSc previous one. So in 1 or 2 days, I'm going to finalize the marks of the MSc previous. Then by the end of this week, I will complete your marks also, your class marks also, and definitely will inform you. If anyone have not submitted the assignment, then definitely I'm going to communicate regarding that, or I will display that these are the students who have not submitted, and those who have requested that they have requested some grace time, then I can consider. Otherwise, those who have not able to ask me, those who have not shown any problem with them, then I'm not going to consider their assignment. अगर किसी का पहुंचा है, आपने सबमिट किया है और वो मेरे पास नहीं पहुंचा है, तो आप वो जो आपने पहले भेजा है उसका आप मुझे मेल भेज दोगे कि मैंने इस मेल पे भेजा था, उसमें सब पता चल जाएगा कि आपने कब भेजा है. So no, don't worry about that. ऑल दीज थिंग्स विल बी मैनेज। जब आपने कहीं से कम्युनिकेट किया है तो कुछ तो आपके पास इंफॉर्मेशन होगी, वो आप अगर शो कर दोगे तो आपका कंसिडर कर लिया जाएगा। So don't worry about that. And only the thing is that you have to be concentrate on your studies. This is very much important. And
Okay,
the
sir. Thank you.
last topic left, that is the mitochondria. Inshallah, I will try to complete on Thursday as an extra class. Thank you very much, and all the best for your bright and best future. Allah
Thank
Hafiz.
you, sir. Allah Hafiz.